This presentation is on research into nanoparticles and resistance. I'm Lauren.
I'm Isabella.
I'm Natalie.
And I'm Jalissa.
What are nanoparticles? Nanoparticles are extremely small particles that have at least one dimension in the nanoscale, generally between 1 and 1,000 nanometers in size. They are usually created through natural processes, such as fires. They can also be created through industrial byproducts, meaning they can be unintentionally produced during industrial activities, such as diesel exhaust or combustion particles. They can also be created through engineered nanoparticles, which means they are intentionally designed and created to have specific nanoscale properties for use in technology, medicine, electronics, and others. They can often be used in medicine, electronics, cosmetics, and structural materials. There are some concerns regarding nanoparticles, one being toxicity to the cells and tissues, meaning that they can enter our cells and reach structures interfering with our organelle function. They can also generate reactive oxygen species, disrupt cell division, cause DNA damage, and alter gene expression. It can also have harmful effects on our bodies as they can enter the bloodstream, reaching organs like our heart, lungs, and brain. They can also cause pulmonary inflammation, cardiovascular toxicity, oxidative stress, and endothelin dysfunction. They can also have negative impacts on ecosystems by disrupting microbiomes in humans, animals, and aquatic species. There can also be some unknown long term effects because there is a lack of data on chronic exposure in humans or ecosystems. Nanoparticles are currently being studied to obtain a better understanding of their toxicity, as well as to prevent harmful effects on humans, animals, and ecosystems. They are also being studied for more medical and technological advances.
Okay, so for the research question in this article, it was trying to better understand if E. coli, specifically K12 MG1655 strand, evolves resistance to silver nanoparticles, and if so, what genetic changes are occurring to make that happen. And why we want to understand this is that researchers want to better understand if E. coli can adapt and survive increasing nanoparticle exposure as they're used in everywhere, as previously mentioned. And then we want to determine as well whether the long-term use and exposure to these nanoparticles could no longer be deemed as a safe antimicrobial since they may be potentially building resistance. And then as well, we are researchers are under the assumption that they do not build resistance. So they're trying to better understand whether we can make this assumption or not. Okay, and for the methods, the first thing that they tried to do was create the control. The first thing that they did was make their control and treatment groups. So first they did this by getting the E. coli strand, that was selected to be tested, and they specifically chose this strand since the paucity of the silver and antibiotic resistance loci is known for this specific E. coli strand. And so once this was selected, the E. coli strain was cultured by using DMB, dextrose, and thiamine, hydrochloride. And the total, they cultured a volume of 10 milliliters. And so then after that, the stock culture was propagated by transferring 0.1 milliliters of each of these cultures into DMB daily. And then this helps create, so they did that. And so then the controls and the treatment were both created by doing 5 different 0.1 milliliter samples that were added to 9.9 milliliters of DMB. So then in total, they created 5 control groups and 5 treatment groups. And then once these were created, these treatment groups were exposed to different concentrations of these nanoparticles. So specifically for the first 50 generations, they were exposed to 50,000 nanograms per nanoparticle. Then the next 90 generations were exposed to a higher concentration, and then the next 45 were exposed to even higher concentration. However, they did run into like a quote unquote error, like an experimental twist had occurred, as it was observed that some of the treatment cultures started to die when they were exposed to the highest concentration. So what they did is they created a quote unquote cocktail, and what this cocktail consisted of was mixing the remaining surviving cultures and allowing it to grow for 40 generations in order for the culture to recover. And then after this growth, they were again exposed to the 125,000 nanogram per liter concentration and this helped ensure that the resistant trait was not lost in the treatment groups. And so once those groups were created, the second step was the bacterial growth was measured by using a 98 well plate Synergic MC spectrometer. And then these were measured at different hours, so at hours 0, 3, 6, 12, and 24. And then the third step was to determine the populations. So the populations were determined by plating the samples at 0 hours and 24 hours. And then these samples were diluted. And then also they determined the minimal inhibitory concentration. And this was to help determine the mean concentration of the serial dilutions that they did. And then lastly, the actual gene manipulation analysis was done. And so what they did was they took the generations 100 and 200 from both the controls and treatment. These samples were then grown in media for 24 hours. And then once they were grown, the DNA was extracted and prepared, and then they were observed. And specifically, they observed for single base mutations, any assertions and deletions of the nucleotides, and if any of the DNA fragments were moved within the genomes.
So in terms of the main result, obviously the purpose of this study was to determine how rapidly and what kinds of genomic changes that silver nanoparticle resistance could evolve in E. coli, the K12MG1655 strain, which doesn't harbor any silver resistance elements initially. So in terms of the figures that best describe the main results, figure 5A on the left shows that generation 250 E. coli had superior growth at exposures of 250, 500, and 1,000 micrograms per liter of 10 nanometer citrate-coated silver nanoparticles compared to the control group. And then figure 5B shows that the generation 250 E. coli again showed superior growth to the control starting at about 100 micrograms of the silver nitrate cocktail. And then the treatment cocktail showed less reduction in growth compared to the control cocktail concentrations. And then in terms of the main conclusions, as stated previously, generation 250 was approximately day 38, which shows how fast the E. coli could become highly resistant to silver nanoparticles and ionic silver in a really short period of time. By generation 162, phenotypic evidence was observed that showed silver nanoparticle resistance of the treatment groups greater than that of the control populations. After 225 generations of exposure to the silver nanoparticles, the treatment populations showed greater fitness compared to the control strains as measured by optical density and colony forming units in the presence of both silver-coated nanoparticles and silver nitrate or ionic silver. Genomic analysis also revealed the accumulation of silver nanoparticle resistance, associated changes in the treatment populations by generation 100. And then by generation 200, 3 mutations had reached high frequency in the silver nanoparticle resistant stocks. And so this study indicates that despite previous claims, bacteria can easily and quickly evolve resistance to silver nanoparticles through relatively simple genomic changes. And the results also indicate that extra care should be taken with regards to the use of engineered nanoparticles as biocides, as well as regards to unintentional exposures of microbial communities to nanoparticles in waste products, because as seen before, resistance can evolve very rapidly.
What we found interesting was that bacteria can evolve resistance to silver nanoparticles really fast, even though many people assume nanoparticles would be resistance proof. It clearly shows that evolution is powerful and can happen in real time. It was also interesting how quickly nanotechnology is growing in everyday products, yet safety studies are lagging behind long-term and environmental risks. Some key things are that silver has multiple antimicrobial actions, but a few mutations are enough for E. coli to survive it. Nanoparticles can travel inside the body and interact with cells in unique ways. Current waste treatment systems cannot fully remove nanoparticles, meaning that they can spread into soil and water. Studying safety must include multiple generations because it can affect or gather up over time. And overlap, bacteria exposed to silver nanoparticles that had helpful mutations survived and reproduced, while others died as an example of natural selection. Rapid evolution in microorganisms is a resistance developed in just 200 generations, showing how quickly evolution happens in bacteria. A few small DNA mutations led to major survival advantages is an example of genetic variation. Adaptation to a new environment is related to the nanoparticles acted as a new selective pressure to bacteria had to adjust. Sorry. Seeing an experiment track evolutionary changes made course concepts feel more concrete and understandable, which is an example of evolution in real time. And scientists have to use evolution when it comes to creating tech, like the antimicrobials, which can be a good impact on humans. The end.
That concludes our presentation. Thank you.